I would talk about generation of wine yeasts, particularly intensifying floral aromas in wine. Just to start, I'd like to talk about the institution where I work as researcher for the last 15 years, the Australian Wine Research Institute, the AWRI, based on Adelaide. That's the capital of Southern Australia. It's the 5th most populated city in the country. I have highlighted the state in red. It's double the size of Spain. Even though the distances are huge in Australia, lucky enough, AWRI is only 1 hour time drive from the most important wineries, such as the ones in the northern part of Australia. Also, South Australia produces over the 50% of the wine of the whole country. In 2021, Australia was the 5th wine producer in the world with around 14 million hectoliters. That means 1/3 of the production of Spain. Here you can see the building of the AWRI. Even though this building was inaugurated in 1928, the institution has over 60 years of existence with around 100 employees, and it is financially supported by Wine Australia and the government of Australia. I would also like to mention that we have an plan of research of 8 years. I work on the field of generation of yeasts to improve the quality of wine. As you know, Saccharomyces cerevisiae is the main one used for the making of wine, for the transformation of the must sugars into ethanol and CO2. But at the same time, this yeast creates or produces secondary metabolites such as acid, higher alcohols esters, and CSV. Even though these are in a very small amount, they are very important for the flavor of wine. I would like to highlight that for over 60 years we have developed and isolated strains of yeast in order to provide options for enologists to differentiate their wines and to have non-risk fermentations. So where do these yeasts come from? The most common and traditional method is to isolate dominant strains of wine fermentation, typically a yeast that had produced wine with particularly exceptional sensorial characters. AWRI has right now, currently, a country collection of over 2,000 natural strains being used in the Australian wines. However, this traditional method, is rather limited because it does not have the needed potential to develop new strains that improve the ones that exist already. This is very important because there is a demand by enologists of yeast with a wide range of improved or optimized sensorial features. How can we get these improves? For the past 40 years, certain genetic techniques have been used for the domestication or monitorization or control of vinic or wine yeasts, similar to the ones used in other sectors of agriculture and livestock. Two of them are hybridization and mutagenesis. Any organism produced by these two techniques is considered a no-OMG, non-genetically modified. I will be talking about mutagenesis particularly. Mutagenesis is a natural process by which genetic information of an individual, its DNA, changes and creates a mutation. This is the main strength driving evolution. One of the possible potential consequences is a change in the phenotype or in the observable feature of the organism. A relevant example is the generation of the Pinot Gris from Pinot Noir. The difference in color is thought to be attributable to a natural mutation that happened many centuries ago in the Pinot Noir variety. As in this example, this mutation happened naturally or spontaneously in nature, but at a very low frequency, and that is why we use mutagens in laboratory to accelerate this frequency. You can see here a physical agent of this. An example of physical agent is UV. The dose is critical. We would be applying the common expression, what does not kill you makes you stronger. If we used a higher dose, most of the population would die, and then the survival would show multiple mutations, increasing the chance that the relevant aspect that we need in the enology would be negatively impacted. However, too low a dose would not be enough. That is why it is so important to identify the needed optimal dosage. In general, to create diversity through mutagenesis is a relatively simple process. Later on, we find in our hands a lot of multiple molecules, but we need to apply selection. For that, we need that the strains with the desired feature have a particularly selective advantage. When we apply the selective pressure, only cellules with the desired phenotype will be developed and will grow. I would give you 2 examples on how we applied mutagenesis and selection to generate wine yeasts with sensory characteristics that are improved and that are considered non-genetically modified organisms. First of all, the creation of low yeasts on H2S, and later on I would talk about the generation of yeast that intensify floral aromas in wines. Well, talking about the first example, the presence of H2S is a very negative compound because it is associated with reductive notes and characters with bad smell of rotten eggs. We know that they are responsible for the production of wine yeasts are responsible for this compound. Some years ago, during research between AB BIOTEK and AWRI, we were able to develop yeasts with low H2S. How? Well, we subjected a PDM strain to chemical mutagenesis, and after that, the mutagenetized population was submitted to a biggie agar process. The darker the colony, the bigger the production of this compound. After a cribbage of more than 16,000 colonies, we were able to identify 8 strains with a more clear phenotype than the other strain, as you can see in the image. This phenotype that has hydrogen sulfide in a low content was cultivated in a Riesling variety. As you can see here, the PDM strains created detectable levels of H2S during alcoholic fermentations, whereas in one of the isolated strains created much less of this compound as compared to the PDM control strain. So a few years ago, this strain was commercialized by AB AWRI, and it is recommended for the elaboration of fruity wines, even in musts with a low concentration of nitrogen. And the removal of reductive notes enhances the varietal nuances of the wine. Now, talking about the second example with the overproduction of floral aromas. This is a desirable attribute because it is a trend among consumers that they want fruity and floral wines. One of the compounds that is produced during AF is 2-phenylethanol, or 2-PA, that is linked to the rose or floral aromas, and it is derived mainly from the catabolism of amino acid phenylalanine. And it can be esterified by the yeast to produce the 2-acetate phenylallyl. What is the contribution of these compounds to the floral compounds? Here you can see the difference of the two compounds in two different varieties of wine, red and white. This shows a certain impact of these compounds to the aroma of the wine. The value of the activity of phenylacetyl, of the 2-PA, 2-PEA is really not detectable. But in the case of 2-PE, its level is around 1 for white wines and over 1 for red wines. This graph shows that, in general terms, these compounds are not generated in sufficient amounts by the yeasts to mark the difference, to have a difference in the aroma. So if we are able to create or produce yeasts forming more of these compounds, this could have a significant impact on the aroma of the wine. How can we achieve this? We know that the production of 2-PE depends on the levels of the precursor amino acid phenylalanine. But we also know that strains that are resistant to the toxic byproducts of these 2-PE create much more phenylethanol and the floral aromas. Taking all this in consideration, the commercial strain by AWRI was subjected to mutagenesis, chemical mutagenesis, and then these strains were implemented or inoculated in the physical environment. Also, not treated strains were also implemented with the hope of having spontaneous mutagenesis. After several days, we were able to isolate around 73 resistant strains. These strains were used to ferment a very small volume of a synthetic must, and around 20 of these strains showed very intense aroma at the end of fermentation. These strains were then used to ferment a small amount of a Chardonnay must, and the levels of floral compounds, 2-PE and of PE, were measured at the end of fermentation. This graph here shows the production of both floral compounds by the different isolated strains as compared with the control strain, with a value of 1 in this graph. We can see how, on average, the isolated strains produce around 30 parts over as compared to the control. And in the case of 2-PE, the increase is a bit smaller, around 6 times than the control strain. Taking all this into consideration, initially we isolated one of these strains called AWRI 2940. I would call it Rosa 1 from now on. And this was decided because it really created these floral compounds. So Rosa 1 is used for vinification of 20 liters of a Chardonnay must, and its performance during fermentation is compared to the control strains 796. These vinifications were done at 15 degrees in triplicate. This is the first practical cases I will be sharing with you. As shown in this graphic, this Rosa 1 strains in blue fermented the sugars in the most at a similar speed as the control strain, and the AF was completed after 15 days. I didn't get the time at 15 degrees. And then it was completed before the bottling of the wines. As you can see, the control strains created lower or below the threshold of 2 PEA, whereas with the fermentation with the Rose 1 strains resulted in in 37 times the levels, and also 7 times more of 2 PE as compared with the control strain. About other byproducts of the fermentation, Rosa 1 created a level of esters that were higher than the control strain. Certain esters related to fruity aromas such as banana and apple. Also, the fermentation with Rosa 1 did not result in any increase in the levels of acetate ethyl connected to negative characteristics such as nail lacquer. It was also studied the evolution of concentrations of floral compounds with the time, the bottling time. As you can see in this graph, the concentration of 2 PE was rather stable over time, and concentration was more or less the same in wines even after 12 months of bottling. The situation is rather different. When we see the 2 PEA, as you can see here, the concentration was reduced drastically over time in bottle, and you can see how wines fermented with Rosa 1 really is much higher even after 12 months in the bottle. These results show that these aromas tend to open over time in bottle. After 6 months in bottle, we carried out a sensorial analysis by 10 people panel. As we expected, the floral aroma was the most impacted attribute by Rosa 1, that was really increased, whereas we did not observe any significant differences in other aromatic attributes that were studied in comparison with the control strain. On palate, in mouth, even though Rosa 1 was more astringent and bitter, this wine was not considered negative by the panel, the tasting panel. Now, the second practical case that was also carried out on Chardonnay. We wanted to find out what was the impact of different concentrations of 2 PEA on the aromas. We selected 3 strains. Rosa 1, I talked about it before, producing very high levels of 2 PE. Rosa 2, also producing very high levels of this floral compound, and then Rosa 3 that leads to less high levels of PE. After fermentation, these wines were bottled, and we carried out a thorough sensorial analysis with 10 tasters, and you can see here the results of the sensorial testing. You can see here how the strains are organized from lower to higher production of levels. Those who produced higher levels are the green, red, and rose ones, with a tendency of aromatic and floral, as compared to the green one, which was the control strain. In the right-hand graph, you see how the intensity of the floral aroma is correlated directly with the production of 2PE, and the higher the levels, the more floral the wine. However, if we focus on a negative attribute such as the potato or baked vegetable aroma or smell, we see how these two bad smells is less concentrated in the strains that were or wines that were injected with these strains, and with wines fermented with Rosa 2 strains and Rosa 1, concentrations of methanethiol and methional were significantly lower in comparison with the control wine in red, in green. That might explain why these wines have less reductive notes. Alternatively, this could help mask these negative aromas. Now, in the last practical case that I'd like to talk about, we wanted to study what the impact was of overproduction of 2PE in the aromatic profile of different wines. So we fermented 50 kilos of Shiraz in triplicate with a controlled strain and Rosa 1 strain. Here we can see a summary of the sensory analysis. As seen before with Chardonnay, this Rosa 1 strain in blue created wines with higher aromatic characters or more intense aromatic characters, and also wines fermented with Rosa 1 created a very intense aroma. On the other hand, the notes of red berries and red fruits were masked, and this resulted in wines that were considered less balanced than was expected on a red wine. This graph summarizes the production of 2PE by Rosa 1 and Rosa 2 in the different vinifications over 5 harvests. And you will see how in all the varieties of studied wines, whether it's Chardonnay, white wines or red wines or sparkling wines, these strains produce very high levels of methyl ethanol. On average, these strains produce around 25 times more than in the control and in rosés, and 12 times more in red wines, and 5 times more in white wines. Thanks to the sensory analysis, we were able to determine that what types of wines are more compatible with these floral aromas.
These aromas seem to work rather well in the different varieties of white wine studied, such as Riesling, Chardonnay, or Pinot Gris. We've also obtained results that are very positive in Garnacha rosés. On the contrary, we haven't seen that these aromas are compatible with Shiraz or Tempranillo. And at the moment, we are studying what would be the effect of these floral aromas in sparkling wines with Chardonnay. And the results, preliminary results, are very promising. In order to finish this presentation, I'd like to speak about the genetic analysis that we've carried out in some of the strains where the phenolethanol is produced with the goal of finding out which mutations in the DNA are responsible for these aromas. We've sequenced the full genome of these strains, and we've seen that mutations are concentrated in 2 genes that are involved in the synthesis of the phenylalanine amino acid, which is the precursor of this ethanol. These genes are called as tyrosina 1 and ARO4. In order to prove whether these mutations give out as a result a change in the ethanol content, we've use the CRISPR-Cas9 genetic sequencing technique, which allows us to introduce these changes in an individualized way in a strain called AWRI 1631. Once these strains were used, they were used to ferment a small volume, and these levels were measured and compared between the control strain and the mutagenized strains. The mutations in RO4 in green gave out an overproduction multiplied by 20 times of this ethanol. In the case of, compared to control, of course, and the case of the other strains, in the case of the other trypty character or gene, we saw the in tyrosina 1, they produced over 40 times this type of phenol, and therefore these genes are responsible for the floral aroma. So therefore, summing up, we've isolated a series of wine yeasts that give out floral aroma during alcoholic fermentation. These yeasts have a potential to provide enologists with tools to produce more intense floral aromas, and these tools have been commercialized under the name of AWRI. Thank you very much.